goes in, which I'll write in purple. What comes out, I'll write in green. And then tracking the carbon, which I'll do in black. Now, carbon is going to be something that comes out, so it gets a bit confusing because do I do it in green or do it in black? I'm doing it in black, track the carbon, okay? You want to pay attention to the number of molecules, the nature of the molecule, and the function of the process I'm talking about, okay? So there are really 6 things to keep track of. The names of the molecules, what goes in, what comes out in carbon, and then function. What is the purpose of the process, okay? So those, let's say 5 things to really pay attention to with the process. Leslie will record this, and then it will go onto YouTube, and then it will go into Canvas. So you'll be able to sit and watch this as many times as you like and show your family and your kids and stuff, which is that exciting. Okay. Right, we're going to break it down into 4 processes. You've read about this in your book. First one, glycolysis. Okay, second one is the oxidation of pyruvate to acetyl-CoA. Sorry, there's no simple name for that. Krebs cycle and oxidative phosphorylation. So you've got to track 5 things in 4 processes. When you're studying, break it down into the 4 processes and then track these 5 things. Make sense? There's a strategy for doing this. Okay, there's a simple way to do this well, but there are lots of ways to do it badly. Okay, so what goes in? Glycolysis, glyco, what are we going to start with? Glucose. We're going to start with basically a simple sugar, glucose. So I'll do that in purple. So here's glucose. Glycolysis has 10 reactions. Okay, I'm going to draw arrows and stuff in random colors. So there are 10 reactions. You don't need to know the 10 reactions or the 10 intermediates. We talked about the first one. Glycolysis converts 1 molecule of glucose into 2 molecules of something called pyruvate. Don't have to know the structure. That arrow is 10 reactions. You don't have to know the individual reactions. So, what goes in? Glucose. What comes out? Pyruvate. That's already my mistake, so I'm going to put that in green. You obviously don't need to worry about these colors because you can redraw this with color pens and stuff when you get home. Okay, so that's what goes in. That's what comes out. In terms of at the end, let's think about the carbon. How many carbons in glucose? 6. Someone said over here said a number. 6. Yeah, good. I just didn't hear you. It's not wrong. I just didn't hear you. Okay, 6 carbons. There is no carbon released. There is no carbon dioxide produced in glycolysis. So how many carbons must there be at the end? There must be 6. That's the first law of thermodynamics. But there are 2 of these molecules. So how many carbons in each of these? 3. Okay, so 2 times 3 carbon pyruvates. Okay? There's no carbon that's produced. This whole set of reactions basically serves to do 2 things. One, there's an input of energy so that we can activate the process of glycolysis. And the second one is you oxidize the glucose. I'm already out of breath. You oxidize the glucose. This is my workout for the day. This is like anaerobic cellular respiration for me. Okay, so the point is that you're going to put some ATP in. We'll get a little bit of ATP out, but that's kind of crappy. It doesn't really count for much, but we'll document it anyway. We're going to oxidize the glucose. If we oxidize the glucose, oxidation involves loss of electrons. So electrons are going to come out. Okay, here is one of the most important points about cellular respiration. It is going to be used initially through the first 3 sets of processes. It's going to be used to make a molecule called, I'm going to get the pen colors right. This is what I hate about this. It's going to make a molecule called NADH. Okay, so we're going to get NADH out, and that means we've got to put in a molecule called NAD+. This stands for nicotinamide adenine dinucleotide. It doesn't matter. You can call it NAD+ and NADH. NADH is the reduced form of NAD+. The electrons basically come from glucose and the intermediates, and they're given to NAD+ to make NADH. So, what goes in? NADH, NAD+ goes in. Okay? NADH comes out. So how many? 2. Got to conserve matter, so you've got to balance these things. So we get out of this process 2 NADH. What is the point of NADH? This is a really critical point for this whole kind of spiel. the really critical thing is that NADH carries high energy electrons. That is its function. NADH's function is to carry high energy electrons. This is like, takes me back to when I was in the military. When my boss, my commander was telling me the mission, he would tell me twice, and I would repeat it to him, okay? So, if I repeat something many times, it's super important. Okay, NADH carries high energy electrons. We are basically going to use those electrons down there at the end. So we're going to start accumulating this molecule, okay? We put a little bit of ATP in. So what goes in is a little bit of ATP, and 2 ATP goes in. And if I break ATP down, what do I get out? ADP. ADP, 2 of them, okay? I also get a little bit of ATP out. So I get actually 4. Danger, yes. What comes out? That's green. So I get 4 ATP out of this. If I get 4 ATP out, what must I put in to make ATP? Okay, if I get ADP from hydrolyzing ATP, how do I get back to the ATP? I put in ADP. So we put in 4 ADP. Okay? So that is glycolysis in a nutshell. You can break this down to 10 more complicated sets of processes, but that is a model of what glycolysis does, and it's good enough for us at this point. Biochem will make you memorize all 10 steps. Totally pointless as far as I'm concerned. So, what do we get out? We get out 2 NADH, 4 ATP. That is the functional part as well as the pyruvate. So what is the function of glycolysis? It's to make pyruvate, and it's to make a small amount of NADH and a small amount of ATP. What is my net gain of ATP here? 2, right? I got 4 out, but I put 2 in. My net gain is 2. Yeah? Make sense? It's like money goes into your bank account, money comes out and goes to the bar, and you're left with some for savings at the end. So that's what we're going to put back into savings. What we're really, really interested in is this molecule. Okay, this is the point. That's what we're after. That's glycolysis. Okay, the next thing, what happens to the pyruvate? This is the second step. Pyruvate gets oxidized to a molecule called acetyl-CoA. So, this is the cytoplasm. We're going to take the pyruvate and we are going to move it into the mitochondria. So here is a part of the mitochondrial membrane. That's the inner mitochondrial membrane. Remember that? It's a kind of wiggly membrane. And this is the outer mitochondrial membrane. So let's move the pyruvate in. And when we move the pyruvate into the mitochondria, we get this molecule called acetyl-CoA. Okay? A set of chemical reactions. 3 reactions convert the pyruvate to acetyl-CoA. Now, as we do that conversion, I want you to think about one of these pyruvates at a time. One pyruvate goes in, and we get an acetyl-CoA. As that happens, is what we get is, with the black pen, per pyruvate, we get a carbon dioxide released. So if we start off with a 3-carbon pyruvate, how many carbons are left in the acetyl-CoA that came from the pyruvate? 2. 3 minus 1 is 2. So this is a 2-carbon atom. Okay? That's a complicated atom. The acetyl part is the 2-carbon piece. The CoA is this giant, basically, vitamin-like structure. So we don't worry about this. We worry about this 2-carbon acetyl group. Okay? It's like a methyl group, but it's got 2 carbons if you want to think about it like that. That's what we care about. Move in one pyruvate, a carbon dioxide comes off, and we get this molecule called acetyl-CoA. During that process, we're also going to take 2 NAD+, and we're going to convert them by the oxidation of pyruvate into 2 NADH. Okay? Let me back up. I'm going to make that 1. Sorry. My brain works with 2 of everything for some reason. Come on. You can do it. You can write a 1. There we go. This is per pyruvate. Okay, so one pyruvate comes in, a carbon dioxide comes off, we're left with 2-carbon acetyl-CoA. That is partly due to the reduction of one NAD to NADH. Okay, so this is also an oxidation, but of pyruvate. So, that was for one pyruvate. How many do I have from one glucose? I have 2. So we do the second one, and now we've got 2 carbon dioxides, 2 NADs needed, and 2 NADHs produced. Okay, here is the trick. I don't think there's many tricks about learning biology. Here is one of maybe 3 that I know. And I've already told you one of them before, which is learn principles like homeostasis and stuff like that. When you're trying to study this, think about what happens with one glucose. If you can work out everything with one glucose, you can do it with 1,000,000 glucose, right? There are ratios between all these things. Now, it's like the X-Files. We missed the first 5 minutes. So you can stay and watch the rest of it again. That's cool. Okay, so this is the second stage. This is the oxidation of pyruvate to acetyl-CoA. The first stage is glycolysis. Cytoplasm. This occurs as we move into the mitochondria. That's the second stage. Okay, here's the third stage. The third stage is called the Krebs cycle, or it's got 3 names. The Krebs cycle, the TCA cycle, the tricarboxylic acid cycle, or 4th one is the citric acid cycle. You may see any number of those used in any number of different places that you read. I'm just going to call it the TCA cycle. Okay? It stands for tricarboxylic acid cycle. If you say Krebs, people will know what you're talking about. Krebs was the guy that discovered it. What does the Krebs cycle do? The Krebs cycle is like a furnace. It's going to take this acetyl-CoA and burn it. And when I say burn, biologically and chemically, I mean oxidize. So when people say cellular respiration burns glucose, is what they mean is that glycolysis, the oxidation of pyruvate, and the Krebs cycle oxidize glucose. Okay, so when I say burn, I mean oxidize. Obviously there's no burning per se, but there's oxidation. So, 10 steps in this process. I know none of them. I know none of them because none of them are any use to keep them in my mind. If someone comes to me and says, can we talk about step 7? I get a book out, or I Google it. I don't care that I don't have the 10 steps. Every semester, people memorize the 10 steps. And I never say what happens in step 7 of the Krebs cycle. So that memorization is wasted. Okay, so here, 10 reactions you don't have to worry about. We're going to worry about what goes in, what comes out, and the carbon. What goes in? Acetyl-CoA is what goes in. Okay, it's a cycle, so there's no end. It just keeps going round and round. As long as you're feeding acetyl-CoA in, it keeps turning. Now, we're going to go again to thinking about one acetyl-CoA. We had 2 pyruvates. We converted each to an acetyl-CoA. So here, we've got 2 acetyl-CoAs. I'm going to do 1 turn, and that takes 1 acetyl-CoA. Okay, so per acetyl-CoA is what we're going to do is basically take all of the carbon that we have. Okay? So, how many carbons came from the glucose?
6.
No? In 1 acetyl-CoA, how many carbons from that glucose?
2.
There were 2, right? Some of you said 6, but I lost 2 here. That leaves you with 4, but I've got 2 of these. That means each one must have 2 carbons. You told me that just now because I wrote it down. Okay? So if there are 2 carbons in an acetyl-CoA, and I'm going to oxidize the carbons as we go around the cycle, how many CO2 should I get out? I put in 2. I'm going to oxidize them both. How many should come out in an oxidized form?
0.
2. Okay? So per acetyl-CoA, what comes out? I'll just draw them down here. You can draw them any way you like. 2 CO2. They come out at certain points, but this is a model for showing what goes in and what comes out. So you put them where you like. What's important is that this arrowhead shows something coming out in a direction. A lot of people, when I see them drawing diagrams, do this. They don't put an arrowhead on it. How do I know that you know that this is coming out? I know when you put an arrowhead on it. How do I know that NADH is coming out from NAD+? Because the arrow goes through the reaction and comes out. If you just do this, I don't know that you understand there's directionality here, and that's important. So the arrowhead is super important. So now we're at the citric acid cycle. 2 carbons have come out. I've got no carbon left, right? In my acetyl-CoA, the only carbon is what's left in CoA, and that gets basically thrown back to these processes over here. So, what is the other important molecules? Well, what are the other important molecules? NADH, right? This is what we're trying to make. We've made 2 here. We've made 2 here. Okay? How much ATP have I made so far? 2. What's the point of cellular respiration? At the end of the day, what is the point?
And
The point at the end of the process is to make ATP. The point of these processes is to make NADH. So far, I've not really done very well. Most of my NADH is going to come out of the Krebs cycle. So for each of these acetyl-CoAs, each one that goes in, I'm going to get out, let's go like this, 3 NADH. How many NAD must I reduce? 3, okay? It's just simple balancing things. Okay? Put 3 out, wrong color, purple. I reduce 3 NAD and get 3 NADH. For each of these acetyl-CoAs, I actually get a small amount of ATP out as well. And so, I'm going to get 1 ATP. Now, technically, I don't get an ATP. I actually get a GTP. But it gets converted immediately to ATP. So we're just going to say functionally, you get an ATP. Okay? This is not making ATP, right? I mean, it is. But it's not making what we need. Does anyone know how many we need to get to at the end? A range? Yeah, everyone's like 30, 32, 34, 36. Yeah, it varies. And it varies because of math and because of efficiency. I'm going to say 32. Okay, that's the number I see most commonly. I need to get to 32. I've made 1. Only 2 of those count. I've made 3 so far. This is kind of sucky, yeah? The goal is to make ATP. This sucks. Okay, we've got to have another process, and we'll do that in a moment. So, I get some NADH out. I also get a molecule called, where am I going to put that? I'll put it down here. I also get a molecule called FADH2. And that comes from a molecule called FADH. So I reduce an FADH to an FADH2. It is structurally different from NADH, but functionally, it does the same thing. It carries high energy electrons. So we will need that at the end. This is all per acetyl-CoA. So I do this one time with an acetyl-CoA, and I get these numbers. What if I do it with 2? Because I'm always thinking about 1 glucose. So if I do this twice, how many NAD plus do I need? 6. And I get 6 NADH out. If you ever write on whiteboards, dry stuff before you try and erase it. Okay. We double everything. 2 of those, 2 of those, 2 of those. So, per glucose, 2 pyruvate, 2 acetyl-CoA, they go into the citric acid cycle. It turns once per acetyl-CoA, and that's what we get out. So, the next part is what's going to help us make most of this. Up until this point, these 3 processes in cellular respiration have given us 2 ATP, 2 ATP. I've made 4 in total. I've got to account for 28 ATP. The purpose of this is not to make ATP. The purpose of this is to make NADH and FADH2. I've made 2 FADH2 and 6, 8, 10 NADH. So when I oxidize glucose, the high energy electrons from here, which we need, the high energy electrons from here have ended up in 10 NADH and 2 FADH2. So, there is a whole bunch of chemical potential energy in here, right? We've got to get it out. At the end of these 3 processes, where is most of the energy in that glucose molecule? Technically, yes. Yeah. Which molecules? Let me be more accurate. Which molecules is most of the energy in that glucose in when I've done these three processes? NADH, because there's 10 of them. There's only 4 of them. And there's only 2 of these. Most of the energy at this point is in NADH. And technically, we would say it's generally in these electron carriers. So now the problem is, how do I get the energy out of the NADH and the FADH2? Okay, that's where the third process comes in. The fourth process comes in. The fourth process is called oxidative phosphorylation. Always run, mate. Okay, the third process is called oxidative phosphorylation. So this happens on this inner membrane. So I'm going to draw the inner membrane. Okay, there's the outer membrane, and here's the inner membrane. So I've got to get the energy out of the NADH and the FADH2. So there are 2 parts to oxidative phosphorylation. ATP synthesis and what's called an electron transport chain. So an electron transport chain is a series of proteins which sit in the inner mitochondrial membrane, and there are 4 of them. They will do electron transport. That's what the chain does. And then we have an enzyme which we call ATP synthase, and that's the one which is going to make the ATP. Okay? So how do we get the energy out of NADH and FADH2? Here's what happens. This is called complex 1, this is called complex 2, this is called complex 3, and this is called complex 4. They have long chemical names. One is called NADH dehydrogenase, and one is called cytochrome oxidase. Don't worry, 1, 2, 3, and 4. Is what 1 does is it takes a molecule of NADH and it oxidizes it back to NAD+. The electrons go into complex 1. So this now carries a pair of electrons. The electrons get shuttled from complex 1. There are little transfer proteins which can move through the membrane. And so the electrons get transferred via these shuttles into complex 3. So 2, I'm going to come back to. We go from NADH to NAD+. The electrons go into 1. They go through these shuttle proteins to 3. Then they go in a shuttle protein from 3 to complex 4. So the electrons end up here in complex 4. Right, here's the kicker. Every time you move electrons, they lose some energy. Okay, so every time they make these jumps from 1 to 3 and from 3 to 4, they give up a little bit of energy. They go from a high energy state to a slightly lower energy state. And so that energy is taken by the complex, like complex 1, and it's used to pump protons into this space between the outer and the inner mitochondrial membrane. That's called the intermembrane space. So you pump those protons. Whenever there's a movement of electrons, there's some energy released. So when we go from 2 to 3, when the electrons jump, we can pump some protons. Okay, so we're pumping protons every time the electrons move. Okay, oh, not 2, my bad, sorry. We don't go through 2. Let's just deal with this. Okay, so we're pumping protons. Every time the electrons drop in energy, that energy is used to do some work. This is a third form of active transport. We had primary, secondary, and this is a third form, which I didn't tell you about. It doesn't have a name. This process overall of pumping these protons is called chemiosmosis, because it's effectively the osmosis of a chemical. I didn't come up with a name, don't blame me, it's stupid. So, the electrons move through the chain and they're stuck, and they're over here. If the electrons are stuck here, you can't keep moving new ones through. It's like a roadblock, basically. This is like the off-ramp at our area, and this is I-25. If the off-ramp is closed and blocked up with an accident, can you put cars on up at I-70? Can you keep pumping cars into I-25 up at I-70 if the road is blocked down at the Spear or area off-ramp? You can't. So the electrons here have to be removed. So the electrons come out, and they are used to reduce oxygen to water. Okay, so let's go back to the equation for cellular respiration before we finish that. The equation for cellular respiration is glucose plus oxygen gives you carbon dioxide and water. I've just explained where the matter is transformed in cellular respiration. And I've also started to explain the energy transformation, and I'm finishing it up over here. Think about where the energy and the matter are transformed. So what we end up with at the end of this is some water and this concentrated proton gradient up here, yeah? If you have a gradient of protons up here, high concentration, low concentration, if they can move, they're going to move back into the matrix, which is the space inside here, yeah? If they can, they will. So this is like a channel. It's a special channel. And when it opens, it's always open, protons can flow back down their gradient. So when things flow down their gradient, there's a release of energy. That energy is used to take an ADP and a phosphate, and it makes ATP. This is where you get the other 28 per molecule of glucose. This is relatively straightforward. This confuses people. There are two things, electron transport chain and ATP synthase. Electron transport chain basically extracts the energy from NADH and makes a proton gradient, and ATP synthase uses the gradient to make ATP. What about the FADH2? The FADH2 cannot be used by complex 1. So FADH2 is oxidized by complex 2. And then there are some protons pumped by complex 2 as well. So that's where the FADH is used, or the FADH2 is used, and NADH is used for complex 1. Okay? You will not walk away from here today looking at this and having mastered it. But is what I hope I've done is be able to show you where the energy transformations are and where the matter transformations are, so you can go away and re-watch this. What I would encourage you to do is think about one glucose, how many ATP, how many NADH, how many pyruvate, how many NADH, how many CO2, how much acetyl-CoA, how much carbon dioxide, how much NADH, how much ATP, how much FADH2. And then think about what happens to those molecules in this process. Okay? What I will do on your quiz next week is I will say, okay, 500 molecules of glucose. How many molecules of NADH come out of the oxidation of pyruvate to acetyl-CoA? You get 2 out, 500 go in there, you get 1,000 NADH out, right? I may say to you, the citric acid cycle produces 1,000,000 NADH. That's just the citric acid cycle. How many glucose were oxidized? There's a ratio, yeah? Okay? You can memorize the ratios, or you can learn this as a story. So you may spend 3 to 4 minutes drawing the story out in Monday's quiz, so that you can come back and just answer a lot of the questions very quickly. Okay? Some of the ratios are very easy. 1 glucose, 10 NADH. 1 glucose, 2 FADH2. 1 glucose, net 2 ATP, gross 4. Some of them you can just memorize. The difficult ones are when I say to you, a mole of pyruvate is oxidized to acetyl-CoA. How many ATP were needed to drive glycolysis? There are numerous permutations of this. The numbers are a means to make sure that you've understood the process, because you cannot memorize every single ratio, but you can understand the process and work out the ratios. So the numbers are a means to an end. They're also good for tracking matter, obviously. Okay? Right, I've run way long, so I'm going to have to abbreviate to the next one. So you guys, up you go. Head out.